Hello viewers, welcome to the CEC Gurukul live lectures. If you remember, on the last turn in our lecture, we had discussed the various carbon concentrating mechanisms or the so called alternative mechanisms of photosynthesis, and we had stressed on the Hatch and Slack or the C4 plants, citing various advantages of this type of a metabolism over the conventional Benson-Calvin cycle, that is the C3 cycle. Just to recapitulate, what we had talked about was that there are two types of CO2 concentrating mechanisms, basically which are going to improve the carboxylation efficiency. They would include the biophysical mechanisms because they would have different types of carbon dioxide and bicarbonate pumps at the plasma membrane level, but this is operative mostly in the prokaryotes and the eukaryotic algae and a few aquatic plants. And the more popular ones were the biochemical mechanisms, and that is why not synthesize the 4C compounds before they actually reach the Benson-Calvin cycle, and we could somehow isolate the rubisco from getting too much of oxygen, and they included the Hatch and Slack cycle or the C4 cycle and the Crassulcean acid metabolism, which we are going to discuss in greater details today. There was a recapitulation of the basic anatomy of the C3 and the C4 plant species, their leaves, and we had discussed at great length about the Kranz anatomy, where the bundle sheath cells were being enveloped or encircled by a ring of mesophyll cells, giving the appearance of a wreath or a kranz like appearance. Not only that, there were different types of reactions occurring in both the tissues, so much so that the PEP, which was the alternative enzyme that was employed to trap the atmospheric carbon dioxide, now amplified the carbon dioxide availability. Say, imagine from 130 microliters to the tune of about 2,400 microliters. So that was enormous concentration, making an atmosphere of carbon rich situations near the rubisco, so that the process of oxygenation was minimized and carboxylation was given a leeway. There was a well defined Hatch and Slack cycle which led to the regeneration of the compounds, the 4C compounds, and then back into the mesophyll cells, back from the bundle sheath, but this process was overall a bit costly, and we had reached a conclusion that 30 ATP were required for per C6 sugar as compared to the 18 ATPs which were there in the C3 cycle. Notwithstanding this huge discrepancy, yet we reached a conclusion that C4 plants were much more efficient photosynthetically in terms of productivity, in terms of carbon fixation, because they did not show the so called wasteful process of photorespiration. But have we ever realized what type of life prevails in the wilderness in the form of xerophytes? The conditions are so very drastic in terms of extremely high light intensity, extremely high temperature, and very low moisture conditions. So there's obviously a need to conserve one's stored water, lest the plant gets dehydrated or over transpires to death. So these type of plants which are in the arid zones, they are the succulents, and the cacti, the euphorbias, and many more which we will discuss subsequently, they have evolved different type of strategies to concentrate carbon dioxide, and in turn there are so many selection pressures which have induced the engineering of special type of strategies or of the reactions which are occurring in their tissues. The CAM plants, popularly called as the Crassulcean Acid Metabolism, the name itself is quite misleading, as we would see subsequently. It means what it does not. So they are actually the plants with an alternative carbon fixing pathway, and the basic idea is to conserve water at any cost. In climates where the days are extremely hot, there is extreme desiccation and high temperature. If we look at the beginnings as to how this whole process was recognized for the first time, it was incidentally the French scientist Theodore de Saussure who observed as early as in 1804, and he published a book where he mentioned that the opuntias or cacti, when they were illuminated, that is, they were exposed to light, they could produce oxygen even in the absence of carbon dioxide. Now, this was something which was out of the book and was absolutely against the dictum of photosynthesis, because we have throughout been talking about the equivalence of the carbon dioxide and oxygen being the raw materials and the products respectively. Work of de Saussure was finally confirmed by other scientists like Aubert as late as in 1892, who also published his results in his book, and Richards in 1915 had a chapter by the name Acidity and Gas Interchange in Cacti, published by the Carnegie Institution, where he had specially mentioned that the work of Saussure was experimentally proven. But then nobody knew as to what was the mechanism or the physiological basis of the entire process. A very interesting story goes by an Englishman, Benjamin Hayne, who incidentally was staying in India for about 2 years in 1812-13, and he made some very interesting observations regarding these plants. We could also do it ourselves. He found that the leaves of the ornamental plant, the common garden grown plant, Bryophyllum calicinum, which is a member of Crassulacea, they are all succulent plants, and these plants were growing in his garden, and when he tasted them, he found that they tasted different during mornings and afternoons. Now, this was a very interesting situation, because the leaves had a normal taste just like any herb would do in the afternoon, but if chewed in the morning, the taste was very acidic. So that means something was going on in terms of physiology, which was resulting in perhaps an accumulation of an acid. His observations were very acute, but then there was no scientific basis attached to it, although he even reported these observations through a letter which he sent to the Linnean Society in 1813, but nothing came out of that. We should remind you, viewers, that the taste differences, that is very increasing sour taste during night, now becomes sweet during day. So perhaps it is the accumulation of malic acid in the vacuoles during the night, and this whole malic acid is being utilized or used up during the day. So we will build up our theme over what is called as acidification during night by these plants and de-acidification of this malic acid during daytime, which makes a very interesting discussion. The term CAM or Crassulcean Acid Metabolism was coined by Ranson and Thomas in 1940 because most of the work was done in members of the family Crassulacea, which includes Crassula, which is the jade plant, and sedum, which is the stonewort, which is a lithophyte. but the name itself is a bit awesome. The Crassulacea, as we know, is a stone crop family, and it is actually an Orpine family, and there are a lot of dicot flowering plants. But the main characteristic of this family happens to be the presence of succulent leaves. Although their distribution is worldwide, but most of the members of Crassulacea, they are localized to the northern hemisphere and southern Africa. Typically the dry, very hot or even extremely cold areas. The underlying fact is that the water is scarce, surprisingly, although a few of the species are even aquatic. So a CAM plant which is now used as an ornamental in our houses, the jade plant or crassula, with very beautiful thick leathery succulent leaves which are water filled, and the sedum plant which is a stone crop which we grow in our garden or even in our drawing rooms as a stone crop, and we add a xeric touch to it. The name CAM actually refers to the acid metabolism occurring in the family Crassulacea, and not the metabolism of Crassulcean acid as the name suggests in the first glance. Actually, there is nothing like Crassulcean acid. There is no acid by the name Crassulcean acid. So, it is a non-existent chemical entity. So, what we really want to say is that here is a metabolism which is involving an acid and which is occurring primarily in members of the family Crassulacea, although there are many other families, as we would see. The basic idea of CAM metabolism is that it occurs in very hot and dry conditions. So, they are all stressful conditions for photosynthesis. The availability of water could be purely seasonal, and all these plants are subjected to more frequently than normal to severe water stress. CAM plants are seen in cacti, succulents, and even in many epiphytes, and there are many families. It has been calculated that over 16,000 species are exhibiting CAM cycle or CAM metabolism. They include the family Cactaceae, Crassulacea, the bromeliads or bromeliaceae, which is also having pineapple, Cyperaceae, the sage family of monocots, Euphorbiaceae. We have all seen the giant euphorbias of Mexico and the other deserts, Orchidaceae, which are primarily the epiphytes, Portulaceae and Vitaceae. That is the family which includes other epiphytes like grapes. CAM plants are represented surprisingly by even gymnosperm families. For example, Zamiaceae and Welwitschiaceae, which is a member of Leetails, and more so to the lower ones like the pteridophytes. For example, family Isotaceae, Polypodiaceae and Viteraceae. Now, the occurrence of this type of a cycle in angiosperms and in gymnosperms and the vasculocryptogams tends one to believe that perhaps this type of metabolism originated earlier than even the C4 plants, and perhaps the CAM metabolism is of independent evolutionary origin, although many more genetical studies and genomic studies are required to prove this point and the entire trajectory of evolution of photosynthesis. So, the famous agave plants which are highly succulent in their leaves, a very popular plant for medicinal purposes today, and the pineapple or the bromeliads, they are all CAM plants. Whether they are obligate or facultative, we would see in the classification. And the famous cacti with all the different types of phylloclades and different types of adaptations by which the stems are reduced and they are green and photosynthetic. So, all these plants, they reveal one common point, and that is that somehow Crassulcean acid metabolism is an adaptation to arid conditions, and it is restricted to xerophytes, and actually such plants do have typical xerophytic characters like thick reduced leaves, as I said, even phylloclades, and there is a low surface area to volume ratio. So the whole idea is to minimize transpiration, thick cuticle, the sunken stomata into the pits. So all typical xerophytic adaptations. Some of these plants may even shed their leaves during the dry season to avoid excessive dehydration and excessive transpiration, or if they cannot, they would now store water in their vacuoles and they would turn succulent and watery. If one looks at a cross section of a CAM cycle, one finds that there is a very thick cuticle, the sunken stomats and the vascular bundles are quite compact and not as luxuriantly occurring in space as do the C3 plants anatomy. So that means everything is it seems to be a bit stressful or under stress. So all these anatomical adaptations that these plants are showing is basically to minimize water loss and somehow avoid transpiration and photorespiration. So such type of plants have evolved a particular type of or a unique type of an engineering. It is due to the stresses and the selection pressures in nature that now their entire diurnal rhythm has changed. In fact, it has been reversed. The stomats are going to open during night and they are going to be closed during daytime. This happens to be opposite to what a child is always taught. So these are not the photoactive stomats, but their behavior is scotoactive. This type of an idea emerged only in the 1970s, and before that we had no idea of this type of a diurnal rhythm of plants. So the basic idea is to isolate the tissue of the plant from the atmospheric gases. So this would be some sort of a temporal isolation, that is, some reactions are going to take place at night, for example the C4 cycle, and the C3 cycle would occur during day. So now this temporal isolation which is found in the CAM plants is different from the one which we discussed on the last turn in C4 plants, because then it was spatial and both the reactions were occurring simultaneously. Let us look at the mechanism of the whole process of CAM cycle. Basically, it is nocturnal, that is at night, the carbon fixation. So the first step would be that in the plastid or in the chloroplast which had made starch through photosynthesis, starch is gradually being broken down into the triose phosphates, and ultimately a 3-carbon compound, that is phosphoenol pyruvate, is formed. And this phosphoenol pyruvate now leaves the chloroplast and enters the cytosol. This entire gambit of reactions is taking place in the cell at night. So it is a nocturnal fixation. How does the fixation take place? In the second step, this 3-carbon compound, namely phosphoenol pyruvate, it is going to combine with bicarbonate ions. If you remember, the bicarbonate ions are the ones which are taken up. The CO2 is taken up from the atmosphere, and if you remember, it is the famous enzyme carbonic anhydrase which has such a high turnover number. It converts it into the bicarbonates, and immediately then the bicarbonates are also contributed by the carbon dioxide which is being released from mitochondria as a result of respiration, and this process is going to take place during day and night alike. And in the cytosol, when bicarbonate ions are formed, it is this combination of bicarbonate ions and the 3 carbon compound phosphoenol pyruvate which combine under the aegis of the enzyme PEP carboxylase, which is the key enzyme here, and this is the dark fixation of carbon dioxide. This process is essentially the same as we studied in case of the Hatch and Slack cycling. And as a result, a molecule of phosphate is released, and a 4 carbon compound, namely oxaloacetate, is formed. So all these set of reactions are taking place in the cytosol, but outside the chloroplast. In the third step, this 4 carbon acid, that is OAA, is now reduced to malate. And this type of reaction would definitely involve the participation of NAD dependent malate dehydrogenase. It is a dehydrogenation reaction, so that means a molecule of NADH is now oxidized to NAD, and the addition of H now gives rise to malate, and malate enters the vacuole. There is a constant accumulation of malic acid in the vacuole, and this malate is actually actively pumped. That means it requires the expenditure of ATP, and as a result of a continuous accumulation of malic acid, we would find that the acidity of the vacuole builds up at night as the reaction proceeds, and the whole night there is acidification. So much is the degree of acidification that the early morning pH of the cell sap could be as much as 3, which is extremely acidic. If you were to perform a very small game, and if you were to just munch a leaf from a succulent early morning, you will be in trouble, because the pH 3 means virtually, if not concentrated sulfuric acid, something nearby, which can even paralyze your tongue for a few hours. It's so highly acidic. I have tried it once during my student days. So this is the overall picture of what happens during night. The stomats are open, carbon dioxide can enter, the transpiration can take place, and the bicarbonates are formed, and PP carboxylase acts, and there's a continuous acidification resulting in the formation of malic acid. All these set of reactions are actually preparations for tomorrow, that is the next day, which is going to be extremely stressful. I may add here, viewers, that if we are talking of a desert, there is no day life in a desert, because the conditions are so stressful, nothing moves much. It's only that you have a night life in a desert, because the moment the sun is off, then only the reptiles and all these animals, they would come out, and the plant also would open its stomats, that is the CAM plants, during night itself. Most of the malic acid is actually present in a undissociated form. So that means we could ask ourselves a question: so much of malic acid in a vacuole, does it not affect the osmotic pressure of the cell? Is the water relations not affected? The answer is no, not much, because we find that even though the contents are very acidic, it is in an undissociated form. So that means it would not be osmotically active. And the ATP would come, as I said before, and we have shown in the diagram, it is coming from the oxidative phosphorylation of the mitochondria itself. Now, during daytime, a different story ensues. During daytime, as I said, there's so much of light intensity, so high is the temperature that it is nothing but stress. If the stomachs would remain open during this time, the plant would get over transpired and it would get dehydrated to death. So the best thing is to close the stomachs. And if you have read a bit of the mechanism of opening and closing of stomachs, you would be familiar about the modern ABA trigger theory, which initiates the entire process. So once the stomachs are closed, there is no contact with the outside atmosphere in terms of gases. So the next morning, what would happen when the stomachs close? This malic acid is now transferred, or it is transported out of the vacuole into the cytosol. Now once this malate is into the cytosol, it has to be decarboxylated, because this is daytime, and it will get decarboxylated to pyruvate by now entering the chloroplast, where now the enzyme NADP malic enzyme is going to break down this 4 carbon compound into carbon dioxide. So 1 carbon gone and a 3 carbon pyruvate, which you know is the basis for the formation of starch. Now this is a process which is occurring. Meanwhile, the carbon dioxide which has been released as a result of the activity of NADP malic enzyme is now going to get assimilated by Rubisco in the form of Calvin cycle. So that means there were actually 2 pickup points for carbon dioxide. However, these 2 carbon fixations took place not simultaneously, as was the case in the C4 plants. They took place in day and night respectively. So that means the 3 carbons have given rise to sucrose and ultimately to starch as the reserve food material. The malate level is very high, which was very high before sunrise, gradually is going to be lost, and the carbohydrate levels which were lowest at that time would now be increasing subsequently. So that means the acids get decarboxylated gradually during daytime and the carbohydrates are formed. The process of photosynthesis has also begun, and some of the starch which was built up during the day is now converted at night to PEP to facilitate the formation of malic acid. So the overall picture suggests that the processes occurring during night are different, and during daytime it is a case of de-acidification. If one was to compare all the 3 cycles, one would find a striking point that the Calvin cycle has to run in any of the cases, in all the cases. That means ultimately the sugars are going to be formed by Calvin cycle itself. Now the question is, how does one go about feeding carbon dioxide into this cycle? Whether it would come through a spatial isolation or it would come through a temporal isolation, as we just saw in details in the form of CAM cycle. The point is that this type of reciprocal diurnal fluctuation of malic acid and the stored sugars would take place during night and day respectively, and the 2 events are temporally separated, whereas in C4 plants, as we have repeatedly said, they are spatially isolated. Why we are stressing this distinction many times is that in terms of evolution, we have to first understand whether both these mechanisms they originated simultaneously, or whether it was the engineering of nature in lieu of, say, giving out some other aspect, in the sense that is the productivity had to be put at stake, it had to be compromised, and then only the CAM cycle has emerged, because then the situation was to somehow get away from stress. So, to summarize, the C3 and the CAM and the C4 cycles, they have the same type of job to do, but at different levels. One does it simultaneously but in different chambers, and the other does it in the same chamber but at different times. So that means this is the basic difference between temporal and the C4 cycle, which is just spatial. Have we satisfied ourselves with the question that how and why in the first place, or that is what was the compulsion which led to such a type of an engineering in nature of the CAM plants? We would definitely try to answer this question by saying that the CAM plants have a main aim to conserve water in the arid climates at any cost, because the days are very hot, and the main purpose is to reduce evapotranspiration, otherwise and also grow in such environments which would otherwise be too nasty for them to grow. So these plants have to be extremely water efficient. If one compares the efficiency of water, and the parameter that we use is WUE, that is water use efficiency, we reach a conclusion that if a CAM plant is going to lose, say, about 50 to 100 grams of water per gram of carbon dioxide gain, it is going to be 3 times that in a C4 plant, and nearly 6 times in that of a C3 plant. So C3 plants are going to lose an enormous amount, because they may not have much modifications or adaptations to close their stomats during daytime fully. So that means the CAM plants would have the highest WUE. So an average C3 plant would have 6 times less of WE, and the highest value is of the CAM plants. So this is a unique ecophysiology which has developed, and can we use this unique engineering of nature, the unique ecophysiology of CAM plants for economic viability as crops? Because we know that if we can grow some of the crops or the high yielding crops in situations where the temperature is high and somehow they can conserve water, the productivity can be increased. If one looks at the statistics, we find that the CAM plants are always growing under duress. They may not be as efficient as C3 or C4 plants. Nevertheless, they are the best, or they are the most ideally suited for water deficient conditions, because 97% of water can be lost by the C3 plants, and they have to pay a very high cost of their productivity. The CAM plants can save all of them. Comes a very interesting aspect now that we have highlighted always the arid conditions as being the key word or the dictum for a CAM plant. Nature has its own pathways. There are some submerged aquatic plants also, for example, Isoetis, which is popularly called as a quillwort. They have evolved CAM photosynthetic pathway. They are submerged aquatic plants. So there is water and water everywhere. So there is no question of aridity. And there are so many aquatic plants that we know now, at least 4 genera are confirmed: Isoetis, Crassula, Littorella, Sagittaria, and people suspect that even Vallisneria, which can come out and be submerged, is also an aquatic CAM plant. So their physiology shows that they have a similar nocturnal acid accumulation, that is acidification, and daytime deacidification, as is shown by the terrestrial CAM species, as we have discussed. But then these particular plants have a different reason to have CAM cycle. The reason is not lack of available water, because as I said before, there is enough water everywhere, but the supply of carbon dioxide is limited. So here carbon dioxide is a limiting factor. You would realize the fact that the fusion of carbon dioxide in water is to the tune of 10,000 times slower than in air. So there will be hardly any CO2 available to these plants. What do? They have to conserve whatever carbon dioxide they are getting, and then they would resort to they would engineer a CAM cycle. So added to this misery is that the pH is highly acidic, and when there is highly acidic, we will find that the inorganic carbon species present in carbon dioxide, there is no available bicarbonate or carbonate supply. So then the metabolism needs to be modified. And what happens that during night photosynthesis stops and all the aquatic life is respiring as it was during daytime, but now there is no photosynthesis and there is more and more carbon dioxide being dissolved in water. So these aquatic plants now they utilize this abundant carbon dioxide because there is hardly any competition from other photosynthetic organisms, and in the bargain the photorespiration also is lowered because there is no photosynthetically generated oxygen. So that means this is how these plants have evolved in a manner that even though they are submerged aquatic plants, they cannot afford to have other photosynthetic pathways like the C3 or the C4 even. There is a very interesting aspect of CAM phenotypes, because we just cannot label every plant as a CAM plant. We have to study in details as to what kind of a CAM plant is it. Is it obligate? Will it ever have a potentiality to change its mode of CO2 concentration if given other environmental conditions? So there is a whole spectrum of the different phenotypes which have been observed. The first category is obligate CAM plants. These plants are obligate in the sense that they have no other choice. They will have only CAM in photosynthesis. So let us say they are pure CAM plants. Although the amount of carbon dioxide that they could accumulate would be variable. So we could name them as strong CAM plants or weak plants just on the basis of the quantum of malic acid accumulated, but they still have no other option. It would be CAM or nothing else. There are certain other plants which can be called as inducible CAMs. We can also call them as facultative CAM plants perhaps, because they have the ability to switch from CAM to either C3 or C4, and this would depend on what are the environmental conditions. So this is actually a very, very interesting aspect of the so-called facultative CAM plants, because if they can move from C3 to C4, don't you think that they are the most potential candidates for gene manipulation? If we could know the genes which are responsible, that maybe we can manipulate and even change these strategies at our will. We will talk about it in the genomics part. To cite some examples, there is a plant Portulacaria ephra, which is also called as a dwarf jade plant. Normally this plant uses C3 fixation, but if there is drought stressed, it can use CAM cycle. Similarly, the popular ornamental plant Portulaca oleracea, which is popularly called as purslane, normally is a C4 plant, but it can also switch to CAM when it is drought stressed. Likewise, an epiphyte Clusia is a CAM plant, but when rooted to the ground, when it was an epiphyte hanging, then it was CAM. But then when it is rooted on the ground, it can shift its metabolism to C3, which is a very interesting aspect. Likewise, Eleucharis is an aquatic plant, which is normally having different types of metabolism in the same plant at the same time. It has terrestrial leaves which are C4 and the submerged leaves are C3. Isoetis itself, which is a pteridophyte, it has CAM leaves when submerged and C3 leaves when emergent. So this high degree of variation tells us that the phylogenetic status and the position in the evolutionary trajectory of CAM is still unknown, and lots of work needs to be done to come to this conclusion. There is still another group of plants, for example, the watered plants of Thellinum calicinum. These plants are called as the CAM cycling plants. Their stomats do not open at night at all, and during day they are already stressed. So the plants instead, what they do is they recycle, which is the carbon dioxide produced by respiration, as well as they store the same carbon dioxide during day. So that means they do not open their stomats. These different types of variations definitely would require a regulation of CAM activity at the highest level. It has now been experimentally proved that PEP carboxylase enzyme is the key enzyme, and its regulation is brought about by phosphorylation, because we know that even if the CAM plant is a temporal isolation, we find that the initial carboxylation and following decarboxylation, they need to be isolated at any cost. And this particular enzyme, that is PEP carboxylase, it exhibits a very differential activity, because this enzyme itself is strongly inhibited by malate, and glucose 6-phosphate is the activator. So in any case, PEP carboxylase has to be regulated. Why? Because we have to ensure that it refixes carbon dioxide which has been released by malate decarboxylation during daytime. So for this, what system has been employed by the regulatory machinery is that the phosphorylation of the serine residue of this enzyme, which is also abbreviated as SER-OP, it yields a form of the enzyme which is active at night. And the enzyme which is active at night is relatively insensitive to malic acid, and it can work. So a very beautiful cyclic regulation occurs where the PEP carboxylase, which was the inactive day form, it gets phosphorylated and the ATP is used and the kinase enzyme is active, and as a result the PEP carboxylase active night form is formed, which is insensitive to malate. It can do its job, and when it has to return and convert into an inactive form, it would be acted upon by phosphatase, and water would be required, and now this enzyme would be inhibited by malate. Viewers, you should recognize that all these modifications, all these engineering in nature have to come for a price, because we are saying that the stomats are closed during daytime. So it would just to stop transpiration and exchange of gases. When there is no exchange of gases during daytime, light reaction is taking place during daytime. What would happen to the photosynthetic oxygen which has evolved? And the evolution of photosynthetic oxygen is higher than respiration and other oxygen consuming reactions, because the whole metabolism is going at a very slow ebb, and the accumulation of this oxygen itself may result in the formation of superoxides and other harmful free radical species, which are going to result in detrimental effects on the membrane and even on the composition of the chlorophyll itself. So, to avoid the formation of free radicals, photorespiration has to occur in the CAM plants. So, this is the price a CAM plant pays, and if it does, then naturally the productivity would go poor, and through photorespiration 3 molecules of oxygen will be consumed and only 1 molecule of carbon dioxide would be released. There are various views regarding the evolution or the time scale of a CAM cycle. It is believed that perhaps this cycle could have originated say about 35 million years ago to capture atmospheric carbon dioxide and even scavenge respiratory carbon dioxide in severe arid environments. And nevertheless, it appears to be a case of convergent evolution, but which has taken place many times over. A very interesting aspect is of CAM genomics, which is now having the center stage for various geneticists, because surprisingly 50 angiosperms have been completely sequenced for their genome. And surprisingly, no CAM plant has been completely sequenced, although the dwarf genomes of some CAM species are now available: agave, ananas, kalenko, and the most important of them is Mesembryanthemum crystallinum, and they are in progress. Now, it is very important for us to know and to determine how these key genes involved in photosynthesis and other genes are important to CAM cycle vary between CAM, C3 and C4. This is going to help us to have the whole gene sequences from all the 3 photosynthetic types, so that we can compare them in terms of molecular evolution, what are the structural rearrangements, copy number variations of these photosynthetic genes. Then only manipulation is possible. Recently, it has been found that CAM genomics also seeks to engineer CAM into C3 biofuel food stocks, because we find that the CAM plants have all the necessary qualifications to act as a biofuel feedstock, because a genetics of CAM through comparative genomics is going to be very helpful. And there are several genes which are present, which are CAM specific genes, which have now been characterized, and we know that they are responsible for encoding stomatal and circadian dependent regulatory elements. We also have the help of genomics from the point of view of loss of function and gain of function mutants. That is the candidate genes in kalenko. So it has been now unequivocally realized that phosphoenol pyruvate carboxylase kinase is actually the enzyme which is the key enzyme and necessary for nocturnal carbon fixation and for the normal circadian clock function. So manipulation of this enzyme is going to be very useful. And we know that as the global temperatures are rising continuously, there is a lot of pressure on land. So why not utilize lands which are in regions which are absolutely inhabited so far, and they are hot and dry for typical agricultural crops. why not use CAM plants there? Because as I said, they have all the necessary qualifications. The advantages are that they have reduced water loss, high water use efficiency than both the C3 and C4, and on top of it they have a drought adapted ecophysiology. So that means they have a lot of potential practical applications. For example, they have a very thick cuticular layer. Most of the succulents, they are ideal platform for the development of waxes and even other value added products. What the biofuel feedstocks they require is a high biomass production, and Opuntia and agave, which were hitherto considered to be absolutely useless, are actually having low water and fertilization requirements, but their biomass production is enormous. And on top of it, most of the experiments which are taking place in Arizona deserts, they show that the photochemical and fuel conversion efficiency of CAM plants like agave is enormously high. It is astoundingly 147% greater than that of the C4 species, which itself talks about their potential. And out of these, agave americana appears to be the most productive and cold tolerant species which has been tested, and there is a very strong viability of agave americana as even acting as an ethanol feedstock. What is required for an ethanol biofuel feedstock is that it has a low soluble sugar content, low crystalline cellulose and lignin contents. So as the processes for digestion of these cellulose materials or the raw material into ethanol will improve, Opuntia will definitely become a desirable bioethanol feedstock. And current research is also focused on the potential of inserting certain specific biosynthesis genes into Opuntia and agave americana, so that they can be made into highly priced or highly valuable biofuel feedstocks. So that means the CAM plants of different magnitudes are the nature's engineering marvels in wilderness, and they should not be written off as just the useless prickly pears or the cacti which are dangerous. They have a world of their own. Thank you.